All right, the difference between DNA is that it's double-stranded, whereas RNA is single-stranded. DNA contains the thymine nucleotide, where RNA is the uracil. DNA has deoxyribose as their sugar. RNA has ribose. DNA always stays in the nucleus for the most part, and RNA can leave the nucleus. They're both the same because they're both store genetic information. They both contain nucleotides that are ultimately a phosphate group, some sort of sugar, regardless of the differences, and a nitrogen base. Even though the nitrogen bases are different, this is ATCG and this is AUCG, they both have that base pairing. That's base pairing, is what that says. Where A goes with T in DNA and A goes with U in RNA, this is DNA, and then C goes with G, same thing here. They definitely have the same base pairing. All right, so A right here, this is the phosphate sugar backbone. B, those are the nitrogen bases. C, that's DNA because it's double-stranded, and D, that's RNA. When we make a copy of DNA, it's going to be C, T, G, C, A, G, T. The enzyme that unzips it is helicase. The enzyme that makes it is called DNA polymerase, and the enzyme that bonds it together is called DNA ligase. Oh, looks like I'm going to owe you $1. Now, when we do transcription, DNA replication, we use T's. When we do transcription, we use U's. So this is still talking about DNA replication. So we're still going to have, across from the A's, T's. This is what it, yeah? Lagging and leading strand is because when you have DNA like this, right, here's your DNA, and it unzips like this, right, and here's your DNA helicase, right, that's unzipping it, and you have polymerase. One has to go in this direction, and one has to be added in this direction. So if this is unzipping in that direction, what happens is we have one that's called the lagging strand that's coding in the opposite direction of the helicase, and one that's just following right behind the helicase. That's called the leading strand. And so the one that's lagging has to be done in little chunks, and that's why it's called the lagging strand. Three types of RNA. The transfer RNA is the one that actually has the amino acid onto it, so it looks like this, and it has the anticodon. Remember this, like AUU, right, attached to some amino acid. That's transfer RNA. The messenger RNA is the copy of the gene. It contains the genetic information. And rRNA is located in the ribosome and helps the ribosome attach to the messenger RNA. Transcription is a process where genetic information is copied from DNA to RNA. The enzyme RNA polymerase is what does transcription. And the different types of molecules that can be transcribed are messenger RNA, transfer RNA, and rRNA. All types of RNA are produced through transcription in the nucleus. So now let's look at the difference. If we start off with this as the DNA, then let's say the RNA polymerase comes across to start making the messenger RNA strand. Across from the A is going to be a U, and then still across from T is A's. Everything else is the same. T is A's, but the A's is going to be a U because the uracil is going to be different. And so C, U, A, U, G, U, C. So UAC is our codon, and that's what we're going to use the chart to figure out what amino acid is going to be brought over.